everyone. Today we're going to be focusing on alpha diversity, which measures microbial community structure within a single sample, not between samples or between groups, but within a single individual sample. If you read microbiome papers regularly, you've probably come across alpha diversity. It's widely used, but it's important not to misunderstand or misinterpret what alpha diversity is really showing. And so our goal today is to understand what each metric of alpha diversity is measuring mathematically, what assumptions are there, and then how to basically interpret each type of alpha diversity. Before interpreting diversity, though, we have to think about sequencing depth and rarefaction. So just to review again, that the sequencing depth of a sample, so basically how many sequences you were able to, how many reads of the 16S gene, let's say, were you able to sequence and end up with after filtering, right? If two identical samples, maybe one with 1,000 and one with 5,000, are compared, the one with 5,000 sequencing reads is going to have more ASVs than the one with 1,000. But actually, it really depends on this rarefaction curves. So if you see here, the y-axis is showing the number of ASVs detected, and this is per sample. Every colored line here is a new sample. And then in the x-axis, we're able to look at the differences in sample size, which really means here the number of reads in each sample, or sequencing effort, or sequencing depth, all meaning the same thing. We have a sample here that only reached about 3,000 reads. Not too bad, but compared to the others, it's the lowest, right? And so you could decide to rarefy at this sequencing depth, but it's also too important to consider that there's a couple samples here that haven't plateaued, meaning they're still increasing the number of ASVs with their sequencing depth. And so you have to think about how much you want to sacrifice. Are you willing to sacrifice all of this information as a trade-off to keeping this one sample? Because if you were to rarefy all the way up here, that means you would have to discard this sample. So it depends on what the sample is, if it's critical to the experiment, what the context is. These are some things to consider. Another thing to keep in mind is that rarefaction is important for alpha diversity because alpha diversity is really dependent on richness, which is referring to the number of ASVs or the number of species. But for other downstream microbiome analyses, we can't rarefy. It'll actually distort and bias the data. So what is alpha diversity? It's able to quantify the variety and abundance within a single sample, not across groups, but within a single sample. And so therefore it's able to capture the richness and evenness in an individual microbiome community. And also sometimes phylogenetic breadth, so how related or unrelated the species within a community are. We're going to focus less on this and more on the richness and evenness, but it doesn't tell you which taxa are there. It doesn't tell you the function directly. It doesn't tell you mechanisms, but rather it can be a descriptor that can play an important role, especially in certain microbiomes where diversity is very important. So richness and evenness are two of the main features of diversity. Here we're looking at two communities of species of trees. And what we find is that their richness is the same, meaning how many different species are there, the number of trees, the number of tree species, I mean, between community 1 and community 2 are the same. So we have 4 species found in community 1 and 4 as well in community 2. What's different is their evenness. So what we see is that the distribution of the trees in community 1 are very even. They're roughly similar proportion. But in community 2, we see that 70% of this light green species of tree is dominating. And so we only get 6%, let's say, of this one. And so the distribution, that evenness is much different. One of the metrics we can look at, a very simple one, is the observed species. And so this is looking at simply the number of species, the number of OTUs, the number of ASVs, not really modeling or estimating, but rather just a raw observation. It's in this way intuitive, easy to calculate, but also is very sensitive to that sequencing depth, as I mentioned before, and also doesn't take into account any rare undetected species that other metrics are able to do. And I also wanted to just show here, but this is kind of how this data might be displayed. Other considerations in this particular paper, whenever including the alpha diversity, it needs to be clarified whether rarefaction was performed, how much it was performed, and other ways that the depth might have been controlled. The ACE and Chow indices also attempt to estimate an unseen richness. And so Chow 1 uses singletons that are like species that appear just one time or doubletons that appear twice. And so the correction term is then able to increase the estimated richness accordingly to that. The ACD then expands on this approach by modeling the abundance distribution of rare taxa that have less than 10 counts. In this case, though, both of the metrics are assuming that rare taxa are signaling a hidden diversity, and these estimators can be unstable in very sparse data sets. But if singletons are abundant due to sequencing error or insufficient filtering, the Chow 1 may overestimate the richness. Shannon diversity is one of the most widely used indices, and so this is able to measure the entropy of the species distribution. And this equation is basically taking into account the proportion of individuals belonging to a certain species or species I in this case. And so this is accounting for both the number of species and also how evenly individuals are spread among them. This is taking into account both richness and evenness, okay? Overall, conceptually, this means that it's able to predict the identity. Given that you were randomly selecting one, what are the chances that you were able to correctly predict what species you pulled out of the microbiome bag, right? The theoretical microbiome bag. That's kind of how the equation is working. And so we can take a closer look here at this image, which is comparing different evenness of communities. And so in this case, the richness is not changing, meaning the number of ASVs, species, is not changing. It's going to be 12. Well, what's changing is its distribution. In this first graph right here, all of the species have exactly the same distribution. But as we see, as we go further onto the other graph, that one is much more dominating. This is actually what microbiomes look like more so, because there are typically a few species that are way more abundant. And so what we see here can also be reflected in the graphs, right? We see a huge dominance of this particular species. And so what also follows is that Shannon entropy value. When the Shannon entropy is really high, that means that you don't have a really great chance of being able to predict what species you're pulling out of your theoretical bag here. But here, the Shannon entropy is quite low, much lower than the 2.5 here, which means that you have a higher chance of being able to predict what you might randomly select, in this case, the red one. The Simpson's dominance index is another one that is also widely used. And so this one focuses on dominance and is able to calculate now the probability that if you pull out two species, what are the chances that both of them are going to be the same? So it's different than the Shannon, looking at the probability that both of those species you pull out of the bag is going to be the same. That's what this is focusing on. It's using this equation. And so what's also important to note here is that this squaring of the PI, the proportion of individuals belonging to species I, is squared, meaning if there is any dominance, it's going to amplify that strongly here by squaring. And Another thing to note is how the Simpson's dominance index is displayed. It can be displayed without an inverse and with an inverse. The inverse is much more intuitive because without it, the range is going from 0 to 1. In this case, a higher diversity is lower value, and a higher Simpson's dominance index value here is indicating there's a dominance of a very few species going on. And then you might see the inverse reported. That's more intuitive, where the range is between 1 to infinity, 1 being lower diversity and in the higher values being much higher diversity in that case. This is just a little bit of a summary looking at all the metrics that we identified and went over today, how they are taking into account richness or evenness and dominance. One thing to note is that not a single metric is going to tell you all the dimensions of diversity. So it's important to combine the measurements and think about them all together. So some things to think about, like if richness is dropping, but then the Simpson value is stable, that means that maybe rare taxa are being lost in that case. And the dominance isn't necessarily changing. But if richness is stable and Simpson is drifting, then dominance might be changing in that case. These little distinctions could make a big difference in the biological interpretation. All right, so this is kind of the fun part where we're thinking about, where does alpha diversity play into real life? Well, a lot of different aspects of life. Alpha diversity is often used to link microbiome structure to health and resilience. You might have taken antibiotics, in which case the richness and evenness are frequently disrupted and decreased. And so this allows pathogenic blooms where it can now that it doesn't have a lot of competition, bloom and outgrow and become dominant. That sometimes happens with C. difficile in the gut, for example. In inflammatory bowel disease is another example where the Shannon diversity is often decreased, and this could reflect the ecological instability rather than one pathogen taking hold. There's also in treatments of like fecal microbiome transplants, these are used to restore the richness and Shannon diversity to help potential gastrointestinal symptoms. The microbiome, on the other hand, is interesting because this is a key situation where diversity is actually not favorable. So in the vaginal microbiome, the diversity there is dominated by lactobacillus species. So that is a healthy state that allows an acidic environment that protects the vaginal ecosystem. And so higher diversity there is actually associated with dysbiosis, which means an imbalance of the microbiome. We also have to consider how our microbiomes take hold and increase diversity as we were born, right? And so different routes of birth, whether that's C-section or the natural method, those also can affect the diversity of our microbiome, at least initially, right? Or the evenness. So these are all some things to consider as we think about alpha diversity. But thank you guys so much for listening. And if you have any questions, let us know. But we'll see you next time.